Our next session. I'm Volkan Granit from the University of Miami, and it's my pleasure to introduce Dr. Richard Nowak from Yale, who's going to talk about the B-cell depletion in myasthenia gravis.
Great, thank you, Volkan, and I thank the meeting organizers for inviting me to give this talk. So I'm going to be talking about B-cell depletion in myasthenia mechanisms and randomized controlled trials. These are my relevant disclosures. So this presentation is at a high level, is not meant to be comprehensive, and is not meant to give any treatment recommendations. The main objectives are really to review the rationale for B-cell targeted therapy in myasthenia and review targeted therapy approaches based on the mechanism of action, and also to review the randomized controlled trials and available data to date. It's important to realize that immune cells are key to the development of myasthenia gravis. There's overwhelming evidence in support of the fact that myasthenia gravis is a T-cell dependent, B-cell mediated disease, and that our typical immunosuppressive therapies actually suppress function and or eliminate B cells and T cells. So the mechanisms of MG autoimmunity are quite complex. I wanted to simplify it and talk about some of the basics. So the basic premise is that autoantibodies are produced by a select population of B cells, which result in disease pathogenesis. So there is a strong rationale for B cell depletion therapy. And autoreactive B cells, again, have an established role in the immunopathogenesis of myasthenia gravis and would be reasonable as a targeted therapy strategy in this condition. There's also been a demonstration of the benefit of B-cell directed therapies in other autoimmune conditions, including in multiple sclerosis and in NMOSD. It's important to realize that B cells are not the same throughout their lifetime, and that there is a maturation process. And selecting the right and appropriate B cell for targeted therapy is critically important in terms of treating myasthenia gravis. So one approach in targeting B cells is to do actually B cell depletion. And there's a number of ways to do that. And among those are utilizing anti-CD20 therapies, medications like rituximab, utilizing anti-CD19 therapies, medications like inebilizumab. Anti-CD34 therapies could also be important in terms of targeting select populations of B cells for depletion. So B cell depletion therapy, like I already alluded to and mentioned, not all B cells are created the same, and there are distinct populations of B cells that it's important for us to remind ourselves that what are we doing with B cell targeted or B cell depletion therapy. So CD20 positive B cells are those that are expressing the CD20 antigen on the cell surface. So if you're utilizing an anti-CD20 therapy, you're only depleting B cells that are producing those. It's important to understand what the role of each and every single B cell population is in terms of their function. So I wanted to summarize some key data. So the BEAT-MG study was a phase 2 randomized placebo-controlled clinical trial that primarily was designed to give a go, no-go decision on whether or not an efficacy trial should be pursued in patients with acetylcholine receptor antibody positive generalized myasthenia gravis. The other primary goal of that study was to assess whether or not rituximab was safe in the MG population. There was a lot of concern about PML and other side effects and risks with utilizing B-cell depletion therapy in patients with autoimmune disease in the background of being on immunosuppressive therapy. So what did this study demonstrate? Number one, it demonstrated that, in fact, in this population, again, this is a study that included a total of 52 generalized MG patients, that it was safe. However, we did not see a difference in terms of steroid sparing effects. So the primary outcome was really to assess whether or not there was a difference to give us a go, no-go decision. That said, there were directionally favorable reductions in select MG outcome measures. However, the study wasn't powered to really make any definitive conclusions on those. So again, this study showed no significant steroid-sparing effect difference. And actually, the study did not demonstrate any significant difference in any of the clinical outcome measures. I wanted to point out a couple of limitations of the study. So this is a safety slide. Before I get to the limitations, I wanted to point out that one of the key post hoc analyses when we were doing the safety assessment is that we found that in the placebo group, there was a 3-fold greater risk of MG relapses requiring IVIG or plasmapheresis rescue therapy. Again, this was approaching statistical significance, but did not meet statistical significance. So there might be something more to the anti-CD20 therapy in myasthenia gravis than the study face value actually tells us. One of the study limitations is that the study population enrolled were quite mild. 60% or so of patients had an MGFA class of 2. Prior case series and case reports, in fact, did study patients with more drug-resistant disease, perhaps a greater burden of disease. So we decided to do a subgroup analysis that looked at, again, this is post hoc, those patients that had moderate to severe disease to say, well, maybe we didn't really see a difference in steroid-sparing effect because the patients that we studied were too mild. So we were able to identify 20 patients, 10 in the rituximab group and 10 in the placebo group that had an MGFA class of 3 to 4. And this study is underpowered based on the primary outcome. So we did a test for interaction between treatment group and MGFA class. And here we found that the study population characteristics were well matched. However, we didn't really see a difference in steroid sparing effect. In fact, the placebo group had a pretty high response in terms of their ability to reduce prednisone. We looked at the MG composite and we looked at the QMG scores, again, directionally favorable in terms of the reduction as compared to a placebo, but there was not a difference between those that had mild disease versus those that had moderate to severe disease. Same trends were observed in the ADL and QOL. We also here observed that relapses requiring rescue therapy were greater in the placebo group, but there was not a difference in the moderate to severe versus those with mild disease. I'm going to present some additional data. So one of the considerations additionally was perhaps we didn't follow patients long enough. 52 weeks, was that long enough to actually measure a treatment effect with B-cell depletion therapy utilizing rituximab? So there was a post-study intervention 1-year follow-up period where any patient that enrolled was able to participate. So of the 47 patients that completed Beat MG, 36 of those enrolled in the 1-year extension study. Again, this is not in any treatment. Here, we didn't see that there was any statistical difference in terms of area under the dose time curve in terms of the prednisone at weeks 52 through 96. We also looked at MG-ADL and MG composite score changes. Again, here we have in the blue is in placebo, red is rituximab. Again, directionally favorable longitudinal reductions in ADL and MG composite scores, but none of this met any statistical significance. Some of the limitations here is that these are underpowered analyses. And the other, for the extension study, we did not complete QMG, so those were not available for analysis. This is an interesting representation of MG relapses over the course of 2 years through week 96 in terms of looking at the probability of having a myasthenia gravis relapse. In blue, we have placebo. In red, we have rituximab. So what we're seeing here is that there is a relative risk of relapse in the treatment group adjusted for study time period of 0.33 times that of the placebo group. Basically, in summary, those patients that were in the placebo group had a greater risk of relapse throughout the study period. So that probability was greater. This did not meet statistical significance. I think in part, this could have been underpowered. But again, if you look at this graph, you say, well, isn't there a difference between the two groups? Isn't rituximab potentially protective? And at the end of the day, these are really descriptive and observational data that I don't think you can really hang your hat on necessarily. Again, my focus here is on randomized controlled clinical trials. But I did want to highlight one study that looked at MuSK antibody positive patients. And this was a blinded prospective review that was able to identify a control group as well as a treatment group. And here, there were more favorable outcomes in the rituximab group where minimal manifestation status or better was seen in 77% of patients treated with rituximab as compared to 26% of those in the control group. This is class 4 evidence. Again, this is not randomized controlled trial data, but this is probably the best study we have in terms of evaluating an anti-CD20 therapy in MuSK-MG. So again, getting back to targeted B-cell depletion therapy. So CD19 antigen is expressed more broadly on more B cells. And I think it's important to realize that not all B cells are created equal, like I just said. So anti-CD19 targeted B cell depletion offers broader depletion of B cell subsets that anti-CD20 does not. The key here is that we're directly able to target antibody secreting cells, which are typically plasmablasts and plasma cells. The MINT study, or the myasthenia gravis inebilizumab trial, is doing just that. It's a study that is designed to really look to see if there's a difference in MG-ADL as the primary outcome, enrolling 2 groups of patients, those with AChR and those with MuSK antibody positive myasthenia gravis. So this study is in progress. I wanted to highlight another potential therapeutic in terms of targeting B cells, and that's anti-CD34 targeted therapy. Again, this hasn't really gotten that much attention in the myasthenia gravis community, but this offers us an ability to actually target plasmablasts and plasma cells much more selectively. And if those are actually really important in disease pathogenesis, this might be an important drug therapy in future. Some indirect B cell targeted approaches, again, I just wanted to highlight these. Again, these do not directly deplete B cells, but could have their role. Some of these have been tested in the past, which kind of mixed review, and some of the studies not really achieving their primary outcome measure. So some summary and conclusion. So in clinical practice, we have demonstrated that rituximab is a safe therapy option. However, rituximab does not result in robust clinical benefit as previously has been believed based on case series and case reports. That said, BEAT-MG has not necessarily resulted in a significant paradigm shift for or against the use of anti-CD20 therapy. It just offers us more information about how rituximab is beneficial, and some of the things that have been already previously mentioned is important for us to be able to predict which patients may be responsive or less responsive to such therapeutic approaches. It actually has taught us a little bit about clinical trial design, in that perhaps steroid sparing is not a great primary clinical outcome measure, and one might need to look at things like MG exacerbation rates. So some additional thoughts here. In summary, B-cell directed therapy continues to be a valid target. Again, anti-CD20 is available to us, but looking to anti-CD19 and anti-CD38 therapies, I think, are also something for us to consider. I will point out that there has been a phase 3 trial done of rituximab in antibody-positive generalized myasthenia gravis in Europe. Those data are not yet available, at least not available to me. So I think more information to come once that trial is completed and the data is shared. I think the future of us being able to apply B-cell targeted therapy in myasthenia gravis is where we will have a deep mechanistic understanding of what the roles each of the B-cell subsets play in disease pathogenesis, and which patients might in fact be more or less responsive to said therapies. Future of MG treatment, I think, needs to be evidence-based, mechanism-driven, and individualized to each and every single patient. And so that's where I'll conclude, and that's my final slide, and I'll yield the floor to the next speaker. Thank you.
Dr. Noah. I think we're going to keep the questions for the end of the session. Slide showing. Oops. Go. Thank you. Sorry, I was going to use that as a timer. That's fine. Is this responding? Sorry, I'm trying to move my slides, but no luck yet. Are we able to start? Oops, here we go. I'm unable to control the slides. There we go. Well, I'm Volkan Granit from the University of Miami, and today I'll be talking about the CAR-T cell therapy for myasthenia gravis. And Dr. Nowak did a perfect introduction for my talk because I will be talking about a little bit of B-cell biology to start this. Just on my disclosures, while I did all this work in this presentation in my role in University of Miami, I am currently an employee of Biohaven. However, Biohaven has no role in this study. Okay, so you have seen several similar slides today already that we have achieved a lot in developing drugs to treat myasthenia gravis in different parts of the pathology. But interestingly, we have not done enough, as Dr. Nowak mentioned, to address plasma cells, which are the key drivers of pathology in this antibody-driven disease. There have been a study for bortezomib, which stopped due to lack of recruitment, and TAC-079 is in trials currently. Inebilizumab, as Dr. Nowak mentioned, is expressed in earlier plasma cells, but not in the late differentiated plasma cells. So today I'll talk about a new approach to myasthenia gravis treatment and addressing plasma cell immunology through CAAR-T and the BCMA. So let me introduce the BCMA. B-cell maturation antigen is a very specific antigen expressed really exclusively on plasma cells. The expression starts in early plasma cells and increases as the plasma cell matures. So if we could adequately target BCMA targeting cells, we could selectively be looking at these cells that are the driver of the pathology in myasthenia gravis. So what is CAAR-T? CAAR-T is an autologous CAR-T cell therapy. I'm curious, this is a neurologist audience. How many are you familiar with CAR-T cell therapies? Or, okay, so about half. So I want to describe what CAR-T cell therapies are and talk about complications and why are they relevant. How are they relevant in autoimmune disease? So briefly, CAR-T cell therapies are a newer technology. The technology is taking T cells from a patient through a leukapheresis, and then the selected antigen of target, whatever that might be, in our case, B-cell maturation antigen, is genetically introduced into the T cell. So the T cell now will target our antigen of interest. And once it encounters that target, it will release cytokines and start this immune cascade and kill the target cells. The problem is, so going back, BCMA, so CAR-T cells has had major success in oncology. Really unheard of success rates in B-cell driven diseases with CD9 related disease, and lately in multiple myeloma. So the question comes, why not use this in myasthenia gravis? Well, there's one problem with CAR-T cells, is that traditional CAR-T cells are DNA driven cells. What do I mean? So when the cells are engineered, DNA is introduced into its genome. And then once they encounter the target antigen, they start multiplying and releasing cytokines. So what happens that you get an exponential growth of cytokine release. And this eventually results in potentially major complications. So these are two very specific and typical complications of CAR-T cell therapies. One is cytokine release syndrome, which is characterized by fever as the first step, but if this gets severe, leads to hypotension and hypoxia and can turn fatal. The other is neurotoxicity. So if the T cells damage the blood-brain barrier, it can lead to basically inflammation within the CNS space, leading to headaches, altered mental status, and in advanced cases, seizure, and even cerebral edema and death. This table below is about the two recently approved CAR-T cell therapies in multiple myeloma targeting BCMA, so the antigen that we're talking about. They have impressive success rates, as you see. But what you also see is that CRS, most of them being mild, were seen in most patients. Neurotoxicity was seen in one in 4 patients, about 2 to 3% of these being severe. So if you take these are in refractory multiple myeloma patients. This could be potentially acceptable, but if you propose this in a myasthenia gravis patient, this would probably be not acceptable. So what Cartesian Therapeutics did was, I think, an ingenious way of regenerating these CAR-T cells using RNA instead of DNA. So when you use RNA, when you design your T cells and your T cell encounters the target, it does secrete the cytokines that you want it to secrete, but the T cell and the protein and the RNA has a predicted half-life. If your T cell divides, your RNA is not in the daughter cells. So the activity can be controlled in a drug-like manner. So we could control the dose and response according to how frequently we infuse these patients. So instead of one therapy, like in cancers, we could do repeat infusions, and we could decide how many cells we want to give to a patient and see how much response we can get. So this, in theory, would significantly lower the risk of CRS and neurotoxicity. I'm going to look at my timer. Okay. So Descartes, so Cartesian was first testing this in multiple myeloma and basically compared to what is traditional in multiple myeloma. They have had quite significant improvement in safety. I mean, if you look at these are data in 16 patients, but basically no CRS was seen, no neurotoxicity was seen. So this is unheard of in the field of CAR-T therapeutics. And if those who have seen the CAR-T cell related complications when doing general neurology wards, you better appreciate what I'm talking about with these rates. So about 5 years ago, when I was talking to my colleague, Dr. Kurtol in Cartesian, a classmate of mine from medical school, said, you know, we're dealing with this, new technology addressing plasma cells. I know you're a myasthenia guy. Do you think we can work on this on myasthenia? And we said, why don't we write a protocol? And we went through a lot of iterations, but basically we came up with this, okay, let's give this to a myasthenia gravis patient. Do we have the guts? Well, let's try. We'll start with a low dose and see what happens. So we did a 2-part study. Our first goal was to give ascending doses into patients. So this was a multiple ascending dose, but with an intra-patient dose escalation. So participants received a small dose and a larger dose and a larger dose, and I'll show you more details in the next slide. And then do a part 2 with the maximum tolerated dose, see we can give multiple doses and explore the responses. But our first objective in this first in non-cancer study was safety, tolerability, and then for CAR-T cells, feasibility of manufacturing. How many cells can you isolate from a myasthenia gravis patient and then give them back? What is the maximum dose you can achieve? And then we had secondary objectives of looking at what happens to these patients' MGs, and then exploratory pharmacokinetics, what happened to these cells, et cetera. So just to briefly describe the patient population we selected for the first part, where we were doing the first in non-cancer, we took treatment-resistant and relatively sick patients. So we took patients who were class 3 or 4 with MG-ADL more than 6. We allowed them to be only on prednisone, Cellcept, and azathioprine because we did not want them to be on other B-cell depleting therapies. And at the beginning, we did not allow eculizumab, and Plex and IVIG were not allowed as they may change the immunoglobulins. And they had to have failed at least 2 immunosuppressive therapies for at least a year or have required multiple rescue therapies. And our endpoints were maximum tolerated dose and the frequency of AEs. So this is a schematic of our study. So we gave 3 doses to our patients at increasing levels. So that's 3.5 millions per kilo and 17.5, so 5 times more, and then 3 times more, and 1 week apart. And then we followed the patients through 6 months. So our goal was to give this to at least 9 patients. We initially considered maybe a 3+3 design, but in parallel, Cartesian was running the multiple myeloma study. And what we have seen that the treatment was so well tolerated, we quickly went to part 2. So this was briefly the first 3 participants. They were all class 3A. The medications you're seeing on the right column they were current treatments. They had previously failed multiple drugs. What we have seen that no toxicities, really dose-limiting toxicity occurred, no SAEs occurred, no hint of CRS, no fever or neurotoxicity occurred. We did see some AEs. These were all, since we don't have a placebo group in this open label study, these were potentially related AEs. Basically, one patient got hand numbness for a few days, and the others got, as you see, some muscle soreness, headache, nausea, but these were all really self-limited, not requiring treatment. One SAE that happened was a patient got influenza. I'm reporting this as it was an SAE, but it was felt to be not related to the treatment. See if I can, all right. So the conclusion from this part, although it was 3 participants, in conjunction with the data in multiple myeloma, that there are enough data to move on to the next phase where we could dose more patients. By the way, this was the exploratory analysis of what happened to the patients. So these are only 3 patients. You see their curves. So 2 of them experienced, clinically meaningful benefit, although by 6 months, there was a trend of returning to normal. One of them did not respond, and then they moved on. So in our part 2, what we did was we demonstrated that we are able to achieve a dose of 50 million cells, 52.5 million cells per kilo, and then our patients so far have tolerated it. So we said, okay, what works best? Is it that we need to dose this frequently? Do we have to dose this less frequently? So we designed 3 cohorts, and as you see on the graphs, we have twice weekly, weekly, and monthly cohorts, and follow-up of up to a monthly. So for this part, based on the experience to date, we were more inclusive. We did allow eculizumab as long as beyond 8 weeks. Basically, we did not want that early improvement. We wanted patients to be stable. We did allow patients on MGFA class 2 through 4, not 3 to 4, unlike part 1. And then we did not require the treatment refractory status in this part. And again, our primary endpoint being AEs and maximum tolerated dose, but not maximum manufacturable dose at this stage. So these are our participants briefly, but they happen to be mostly women, 1 male, and you see that the severity was ranged from class 2A to class 4. We also included a seronegative participant and then in the background, we also have some MuSK positive patients who will be enrolled and treated. We aim to treat at least 5 patients per the group, and probably on the weekly group, we're gonna add another 5 patients as seronegative. So to date, so what I'm showing you is really preliminary data up to Friday. We were pushing the sites to get us data, as much as data we can show. The goal was to talk about the design of the study, but I thought we don't want to wait for the next MGFA meeting 3 years from now to show some data. So here are what we have as of Friday. So no dose-limiting toxicities to date, no SAEs occurred, no neurotoxicity occurred to date. Again, this is remarkable for a CAR-T related treatment. Some patients, so specifically 2 patients, did develop fever. So out of 28 infusions that were done to date, 6 events of fevers occurred, 5 of them being grade 1. Grade 1 means above 38, but less than 39 degrees Celsius. And then grade 2 would be more than 39, but less than 40. And all resolved without intervention, although they, per protocol, the patients were admitted, they resolved without intervention. 2 patients had significant headaches. These were, one of them was a chronic migraineur, the other was not, but then the headaches on the non-migraineur resolved. So the conclusion so far in this admitted limited data set is that so far in myasthenia gravis at this dose, CAAR-08, this novel therapy appears to be safe and well tolerated. And this is the exploratory, so what we have clinically, what happened to these people. So we have 2 patients in the bi-weekly dosing, but we don't have their data. They are about to finish their infusions. What I hear verbally is that they have not had a drastic change yet. 1 participant in the monthly dose completed their treatment and they actually dropped off because of lack of benefit. In arm 2, so the weekly dosing, so that which we enrolled 2 patients to date, they had really remarkable improvements. So briefly, this is their composites, like what happened to them within 5 weeks. Again, this is, of course, you know, for when you design and run the study, it's very exciting. Although admittedly, we don't have a placebo comparator, but like a drop of 20 odd points in MG composite or similarly QMG score is encouraging to date. And similarly, the MG-ADL, so all basically outcome measures had a parallel trend of improvement in these limited participants. So basically, where are we right now? So plasma cells, all of, if we could target them, they are an ideal way of, if we could selectively target, of treating myasthenia gravis as the driver of antibody production. Anti-BCMA CAR-T cell therapy, per cancer literature, so per cancer experience, CAR-Ts have been the most effective treatment to address plasma cell pathology, but they're too toxic, as I demonstrated. But the CAAR-08, the mRNA-based, so controlled therapy, may be safe and well tolerated, which the data so far shows, although quite early in the clinical trial. And then to date, if you were to make a go, no-go decision, I think the decision would be a go. This treatment clearly requires further exploration. This study needs to be completed, and then hopefully the encouraging data will be reproduced in the upcoming patients and upcoming weeks. These are my acknowledgements. I actually want to go through these because this study took a lot of development. Dr. Benatar at the University of Miami really helped me, along with my colleagues at Cartesian, design this protocol. Dr. Muzaffar and Howard came into the study later on, but really helped us further the study. And interestingly, the initial design of this study was done at the clinical trials methodology course, where I was back and forth being hit hard on by my mentors at the course. So I acknowledge them for the first part on the protocol development. And thank you for your attention. And our next speaker is Dr. Mario Lozen, and he will be presenting his work on Hinge-Deleted IgG4 blocker therapy for acetylcholine receptor antibody positive myasthenia.
Thank you for the introduction, and thanks to the organizers for inviting me. Not just my work, it's the work of a big team of people. Many of you are actually here, so we have many fruitful collaboration in the years, and I will be showing some of the collaborative effort today. So when we are looking at myasthenia gravis, I think we can learn a lot about the pathophysiology by comparing two forms of the disease. It's MuSK myasthenia versus the acetylcholine receptor form of myasthenia, where the MuSK is predominantly caused by IgG4, although it's interesting to look at IgG1 as well. I think that's important because one of the classical features of myasthenia altogether, I think, is a surprising heterogeneity of the patients, both with respect to the antibodies they have, but also the treatment responses. And this is going to be one of the aspects that I want to talk about today. So of course, it's quite clear that the AChRMG is mostly caused by IgG1, but also IgG3, which is mediating the disease via complement. And as far as we know, there's no evidence that there's substantial complement activation on the neuromuscular junctions of patients, although that might be underestimated because, of course, we don't know about the biopsies in every single person. So that there's still room for work there. So one of the important features of the AChR antibodies that are IgG1 or IgG3 is that the AChR is cross-linked, and we know that the MuSK antibodies are functionally monovalent. And so in MuSK-MG and AChR, there's substantial differences. And we know also that the AChR, usually most antibodies are not functionally blocking the AChR, whereas MuSK-MG, that's entirely different because they're the usual pathophysiological mechanism is that the function of MuSK is blocked. And that's actually, so the work on the more functionally monovalency is the work, for example, but also many other people of Inga Konieczki. We showed that the antibodies are bispecific, as you can see there in the lower left. The antibody has two different heavy chains and two different light chains. So the antibody is both functionally monovalent as well as bispecific, because at any time there's probably only one antigen that will bind to the antibody. In this paper, Mike Huibers and colleagues have very nicely shown that the MuSK antibodies are pathogenic by interfering the binding between MuSK and LRP4. So that was also a very important finding, I think. So we stumbled on the Fab-arm exchange reaction by chance, actually, when we studied IgG4 antibodies against the AChR. And the colleagues in allergies knew this already for years, but to me this was new. IgG4, I just naively injected them into monkeys against the AChR with the idea to study complement activation. But we surprisingly found that the human antibodies in the monkeys were splitting up into half molecules like this and recombining. In this case, the gray antibody could be the monkey IgG4, but this is also a normal mechanism in humans, where there's an interaction between the human and the IgG on the left. So that could be the AChR antibody that are injected. And in this case, the right side, the gray one, is another irrelevant human IgG, or in the case of the monkey study that I did, it was a monkey. In fact, it's quite funny. The monkey antibodies like to stick together with the human antibodies quite stably, so they don't interchange as much later on. And that has led to also a nice way of making stable bispecific IgG4 antibodies that can be used. So the important is that the original antibodies can cross-link antigens, and I'll need this in a future in one of the next slide, whereas the resulting antibodies after Fab-arm exchange are functionally monovalent. So they will not be able to cross-link receptor anymore because they only have one Fab-arm to bind with to the antigen. So there are more differences between AChR-MG and MuSK-MG, and this one is a striking one. So if Jan Verschuren and colleagues with also the group of Isabel Iller have studied the treatment response, and we've also heard about this just in the two presentations before me, that with rituximab treatment, the MuSK patients respond really beautifully. So the MuSK antibody titer goes down substantially in most patients, whereas that is not the case in AChRAb. So there are a few patients included in this study for this subgroup, and you can see the AChR antibody levels fluctuate, and that's typical, but they do not overall go down. Also, the IgG and the IgM levels do not go down with rituximab treatment. So there's and it's of course clear there's not all antibodies are produced by CD20 positive B cells, but of course also plasmablasts and long-lived plasma cells. And that explains the blue line here for IgG and the black line for IgM. But clearly, AChRAb is not responding very well on average, that is, to rituximab, although in individual patients that might be, of course, different. So this is a slide based on data of Professor Mark de Baets in our group, and he's got a very extensive data sets of antibody titers over the period of 10 years in many patients. And what you can see here is that the antibody levels after general immunosuppression, so that's broad spectrum immunosuppression with glucocorticoids and azathioprine, sometimes MMF is using, as well as any other immunosuppressive medication that is needed. The antibody levels do go down over the years, but very, very slowly. You can see that in some patients, even after 10 years, there's substantial antibody titers. So that makes it, of course, hard to study immunosuppressive effects of something like rituximab or prednisone or anything else that's targeting B cells, because you need to wait for the plasma cells to die out, and they can be very long-lived. There are studies in humans that the plasma cells that were generated in the Cold War with a lot of C14 in the atmosphere are still alive in patients these days when they do organ transplantations, for example. So that's quite interesting. So the clinical heterogeneity of the patient with respect to the immunosuppression can also be seen nicely in this slide. Again, these are two patients of Mark de Baets. So the patient with the blue line here had a quite high antibody level, so roughly 80 nanomole per liter to start with. And this patient was treated with general immunosuppression, and the antibody titers dropped rapidly. And this is quite typical for quite a large subgroup of patients. On the other hand, we had this other patient that had the red line that was treated in a similar way and even more aggressively with immunosuppression. And you can see that over the period of 10 years, there was, of course, a substantial reduction of antibody levels, but still at such a high level that this patient was suffering quite substantially from the disease, I understand. So you can see that even after 10 years, the antibody titers are as high as in the other patient after 2 months. So this is a big problem. Of course, this can be explained by the long-lived plasma cells that are existing in these patients. So that's why this treatment that we have so far, based on plasma exchange, IVIG, thymectomy, and all of the broad-spectrum immunosuppressants, were working fine for many patients. For the long term, I think we need more stronger treatments for the treatment-resistant patient, and the last speaker very nicely illustrated how this can be done. Of course, you could look to the long list of other immunosuppressive drugs to address the problem of the treatment resistance. Of course, we need better assay to predict which patient would benefit from which one. I'm not suggesting that any of these drugs are particularly useful for MG patients. It was just to illustrate how difficult the choice would be for a neurologist that had to choose from which drug should I be using. So we definitely need good assays for choosing the right one. Of course, there's some very useful drugs for many patients, but not all of these should be used in combination, at least, or sequentially. That would be very toxic on the long run. So we definitely need better assays for that. So it makes sense to look at the disease pathophysiology that we know about. And we know, of course, that the antibodies are not only produced by B cells, but also by plasma cells. I think predominantly by plasma cells. And that's particularly true for AChRAb, not so much for MuSK-MG, of course, because that responds to rituximab. So the antibodies are produced in high titers, as you see, and they bind to this beautiful structure, which is the neuromuscular junction that I used to illustrate this. So where are these plasma cells? Well, one place, and Professor Nick Wilcox was one of the first people to recognize this, they're, for example, located in the thymus. So this is showing a staining from a patient with follicular hyperplasia. And the brown staining here are individual plasma cells that are found in the lymphoid follicles, so the ectopic lymphoid follicles that are present. So this is one source where these plasma cells can be found. And of course, after thymectomy, if you remove them, you would think, oh, then they're gone, then it should be solved. It's not the case, because they're not obviously not only in the thymus, but also elsewhere, presumably bone marrow, lymph nodes, etc. So we know thymectomy is, of course, very helpful in many people, but it's not solving the problem in quite a substantial subgroup, even if on average it works quite very nicely, I think. So at least for us as a research group, this is a source of plasma cells to study, and we do. So this is one way of addressing it. So we had another drug that was used for multiple myeloma. We used bortezomib, and we used also AChRAb. Both are proteasome inhibitors. They also have the capacity to kill plasma cells. So in this case, we looked at the AChR antibody production in cell cultures. So we take 100,000 cells for thymocytes and culture them for 2 weeks in vitro. And these plasma cells, you can see them there on the right, stained with a green staining for IgG and a red staining for CD138. You can clearly find them back because they produce so much antibody also in the cytoplasm, that's or the ER, I should say, that is then secreted. So we can count them, but that's one thing. You never know what plasma cell you see, of course, when you just look for IgG, but we can also measure the AChR antibodies in cell culture. And you can see from the graph that for the 5 patients that we tested there, they all nicely responded to higher doses of proteasome inhibitors. So these were kindly provided from the biobank of Oxford by Nick Wilcox, except for one patient that was a patient from Maastricht, I believe. So, of course, proteasome inhibitors are one possibility to kill them, and we heard a very nice possibility for the CAAR-T cells. I'm very enthusiastic about this possibility. I put some other options here. I think CD38 is certainly a realistic and easily accessible option because it's clinically already available. There is the possibility of using a CD138 that's currently already limited for because it's coupled to a cytotoxic drug for multiple myeloma, and in principle, anti-SLAMF7 or CS1 is also called CD319 would also be an option in theory. So it could be an option for these treatment-resistant patients as well. And I think it really comes down to the safety profile of these drugs and the efficacy, which one would be the best. I think that is up to future studies to establish. But of course, you would still suppress the entire immune system. So for quite a few years, we've been looking for alternative and safe options. And this takes me back to the idea based on Klaus Teuker's experiment, who investigated which effector functions antibodies have after binding to the AChR. And he used Fab fragments, for example. Also, the group of Socrates Sartos did similar experiments making Fab fragments. And testing the pathophysiology or pathogenicity of them. They were not pathogenic. They didn't block the function of the AChR. And that led to the idea that you can use monovalent antibodies such as Fab-arm exchange AChR antibodies of the Ig4 subtype to prevent complement activation, but also to prevent cross-linking of acetylcholine receptors. And I mentioned at the beginning, this is an important pathophysiological mechanism as well, in addition to complement activation. So importantly, such antibodies that are directed to the AChR in general are not interfering with the binding of acetylcholine. So this acetylcholine receptor, once it's bound to these monovalent antibodies, is actually not functionally impaired. So this activation of the voltage-gated sodium channels will still continue as needed. So there are a couple of options of making different antibodies. We chose to make a Hinge-deleted IgG4 antibody, which is functionally monovalent. It doesn't undergo Fab-arm exchange because the Fab-arm doesn't associate with the second heavy chain at all because the hinge is deleted. And remarkably, this antibody is quite stable. So we use that as a model for our research and as a potential therapeutic tool as well. So what we like to use in our experiment is a human monoclonal antibody from an AChRAb patient, originally isolated at the Scripps Institute in collaboration with Professor Mark de Baas. It's MAP637. Another very useful tool is the MAP35 antibody. That's a rat antibody. 637 is a fully human antibody from a patient. So that's for the antibody. What we can also use are muscles, of course, from patients or from animals. But we like to use in day-to-day research a very useful model, which is a rhabdomyosarcoma cell, which is a stem cell tumor cell line, which is called T671. And Professor David Beeson, who is also in the audience, has made an extremely useful cell line based on this one. It's a super transfected rhabdomyosarcoma cell line that expresses a high level of the epsilon subunit. So you can have the adult form of the acetylcholine receptor in there. So what happens if the antibodies are binding to this, to this TE cell line or to the human neuromuscular junction, is that they will cross-link and internalize the acetylcholine receptor, and then you can measure the remaining acetylcholine receptors on the cells. So this is a model for testing the cross-linking by antigenic modulation. So that's the internalization of the acetylcholine receptor. So you would expect that all antibodies would do that. So this is made possible, of course, because the acetylcholine receptor is highly clustered, especially at the neuromuscular junction, to some degree also in the TE cells, of course, in the T cells, not as much, but in the neuromuscular junction, the acetylcholine receptors are very close together, and that allows cross-linking of receptors by antibodies, but also helps to activate complement because you need 6 IgG molecules bound together to allow C1q binding. So this is an important finding that was already made in the late 1980s. So if we look, what's the mechanism of this cross-linking? There's a very nice work by the group of, or the colleagues of Noritomi et al. They made a model what it looks like. So it appears that the antibodies to the main immunogenic region of the acetylcholine receptor bind in such way that they turn the acetylcholine receptor slightly inwards. They change the angle of the point of attack, so to say, of the acetylcholine receptor. They pull the receptors together, forming these vacuoles, and then these acetylcholine receptors are then internalized. So this is how it works. So for our model system, we use this MAP637, which has a very high affinity to the acetylcholine receptor. John Lindstrom measured it in his lab. It appears to be in the order of 7 picomolar. I know that the pharmaceutical companies were always very happy in the past to have nanomolar affinities. Well, these autoantibodies appear to have even higher affinity by a factor 1,000. So this is actually quite extraordinary. So you need very sensitive assays, of course, to measure such low level, so high affinities. So 7 picomolar is the affinity of MAP637. So based on that, we made this Hinge-deleted IgG4 antibody. So you can see here the sequence. So this core hinge was removed together with the upper hinge. So you essentially get this antibody of IgG637, which is functionally monovalent, like you see here on the right with the green, the structure of the antibody. So it's half of the IgG molecule. And we showed that, of course, as you expect, it prevents antigenic modulation because the monovalent antibody will prevent cross-link of receptors, and that works very nicely in vitro. I'm going to skip over this. But what about the patient sera? So question was, can we use this hinge-deleted IgG4 to prevent antigenic modulation? The first question is, do all sera modulate the acetylcholine receptor? So we had a very nice series of patients from Ana de Rosas and colleague from Pisa, and they sorted them according to the level of antigenic modulation that they caused. So interestingly, all sera caused antigenic modulation, although some of these were borderline positive in the acetylcholine receptor assay. So this is actually also quite a sensitive assay to use. But not all CAAR-T modulate the acetylcholine receptor very strongly. So you can see some that almost remove all of it, they're on the right, and some that only have like 70% modulation on the left level. So when we added the Hinge-Deleted IgG4 antibody, we could substantially increase the AChR levels in most patients, but not all. So to me, that tells me that the antibodies of these patients are also intrinsically very heterogeneous. I think the epitopes that they recognize must be heterogeneous, not only targeting the main immunogenic region, because that's where the AChR is targeted by the 637 antibody. So that's different. So perhaps even different subunits, could be different epitopes within the alpha subunits, but could be also other epitopes. That's also to be considered. So it would be interesting to correlate this to disease severity. I did, it's a partial correlation, but I think we need more parameters like the antibody levels and perhaps the complement activation to make a really comprehensive view of how these antibodies cause the disease and to which degree. Okay, so I was interested in a small experiment to see, can we actually, well, this was preventing the disease in the cell culture, right? So to add the antibodies at the same time and they compete. And now I did a small experiment to see if we can actually recover AChR levels. So this is just the control condition. So if I incubated the cells with the MAP637 after 3 hours, we have more than 50% acetylcholine receptor loss. And we had 2 plasma samples from plasmapheresis patients, patients that required plasmapheresis, so substantially ill patients that we also tested. And they also decreased the AChR levels. So when we added the unibody, so that's the Hinge-Deleted IgG4, that's the UB condition there, we could substantially, in competition with the 637 antibody, as you would expect, normalize the AChR levels. That's not so special, of course, because you have competition to exactly the same epitope. But also these 2 patients that had required the plasmapheresis allowed to normalize the AChR level substantially, not to 100%. But what was interesting to me is that when we added the Hinge-Deleted IgG4 after the modulation was already taking place, so the AChR was gone and the autoantibodies were still there. When we added on the Hinge-Deleted antibody, the AChR levels normalized. So what that means is that the TE cells, the rhabdomyosarcoma cells, have the capacity to restore the AChR levels when they're left alone, so when the modulation doesn't continue to take place. So that gives some hope to the treatment of this type, because in theory, you could allow to the rhabdomyosarcoma cells, or of course the neuromuscular junction, to relax, to stop the continuous attack, and to allow the AChR levels to normalize if you have a treatment like this. So I think that is quite a nice outlook. So we, of course, we're curious, does this work on also in vivo? And we had an animal model, so it's quite a while ago that we already published this. I just briefly wanted to show and summarize the results of that. So we had a study in rhesus monkeys where we injected the MAP637, that's the challenge. So we got disease in rhesus monkey as a result. And then we wanted to see if we could prevent that by giving the Hinge-Deleted IgG4 antibody. So this, there was no obvious muscle weakness observed, but if we tested this by jitter, it was quite interesting actually. The jitter values were extremely high. So this was tested by clinical neurophysiology. So those of you that work in that area will see these jitter values. So these monkeys look completely normal, completely healthy, but they all had extremely high jitter values, telling that they're quite good at hiding the disease. I don't think it's the same for myasthenia patient. I think they will tell if they suffer, but in this social environment of the monkeys, they will try to hide that they're ill, so it wasn't noticed. So we could by giving the blocking antibody, we could substantially reduce the jitter. And you can, you don't actually have to do single fiber EMG. You can even see that in the decrement values. So if you use the decrement values quantitatively and accurately, that can also be done. So that's much less invasive, of course, because you don't need needles for that. So maybe that's something that could be done also. If you do have a good clinical neurophysiologists, the decrement values can also be used quantitatively, perhaps, if it's done carefully. I'm going to skip this in view of time. We also see complement activation in the animals, and that can be reduced by this. And I'm going to summarize my presentation by saying that there's a subgroup of AChR patient that is resistant to conventional treatments, as illustrated by this graph with the antibody levels. So we really need to pay attention to that subgroup, that plasma cells are an important target in this subgroup. And hopefully in the future, we can develop together some antigen-specific therapies, such as maybe these blocking antibodies, or other ideas are welcome, of course. And I think this is a promising development, that we are able to go in the direction of antigen-specific therapies in the future. Thanks so much for your attention.
Our next speaker will be Dr. Amy Payne, and she will talk about MuSK CAAR-T cells, AA.
All right, thank you so much. I'm very excited to be here at my first live MGFA meeting. They're calling the slides up right now. It's great to be able to put some people to names. I've read many of your papers, and I'm really interested to learn from you. It was a great overview from Angela Vincent. So you may be wondering what a professor of dermatology is doing at a myasthenia gravis conference. Hopefully I'll be able to make that clear to you today. Historically, my laboratory is focused on B-cell repertoire profiling in the disease pemphigus vulgaris, and I'll share with you how that led me to MuSK myasthenia gravis. I do have some conflicts at the bottom of the slide. I co-founded Cabaletta Bio, which sponsored this work. So chimeric antigen receptor T-cell therapies, as nicely introduced by Dr. Granite, are a FDA-approved technology for lasting remission of B-cell cancers. There are 4 anti-CD19 CAR-T cell therapies listed at the bottom of the slide that are currently in clinical use for B-cell leukemias and lymphomas. They differ in their co-stimulatory domain as well as their manufacturing process, and there are 2 BCMA CAR-T therapies that are currently approved for clinical use. And as Dr. Granite explained, a CAR or chimeric antigen receptor consists of an antibody as the extracellular domain of the receptor, in this case, an anti-CD19 antibody. And that's linked to co-stimulatory and CD3 zeta activation domains. And when you express that in a patient's T cell, it programs the T cell to kill CD19 expressing B cells. In this case, both healthy as well as leukemic B cells, because all of the B cell lineages express CD19. One of the strengths of a chimeric antigen receptor T cell is that CAR engagement leads to CAR-T proliferation and the generation of memory CAR-T cells that can last potentially for the lifetime of an individual. So this results in pan B-cell depletion, and that B-cell depletion can be lasting in some of the patients for decades, and they require lifelong IVIG infusion to maintain normal immunity. However, this has led to really striking remissions in B-cell leukemias and lymphomas. A subset of these patients are thought to be cured of their otherwise refractory B-cell leukemias and lymphomas. So this really raised the question then, how can these curative cellular immunotherapies be extended to other B-cell mediated diseases? And so I shouldn't say my favorite B-cell mediated disease because MuSK is quickly becoming my second favorite, but pemphigus vulgaris is my clinical and research area of expertise, and it's considered a paradigm for human autoantibody mediated diseases. Here's a patient with blistering of mucous membranes and characteristic histologic findings on biopsy. And the anti-desmoglein 3 antibodies in this disease are 98 to 100% sensitive and specific for disease diagnosis. And in various animal and human skin model assays, they've been shown to be necessary and sufficient for blister formation. So essentially fulfilling Koch's postulates for causation. And corticosteroids and B-cell depletion with rituximab are frontline therapies for pemphigus vulgaris, but in the pivotal clinical trials that led to their approval, it was associated with a 4 to 9% annual rate of serious infections, meaning serious enough to land somebody in the hospital. So as many of the physicians here are aware, really ideally therapy should deplete only the pathogenic autoimmune cells and leave the healthy B cells alone to provide protective immunity. So this has always been the goal, or the holy grail, if you will, of targeted therapy for autoimmunity. So if we think of the B cell lineage markers, again, as Dr. Nowak nicely introduced, the B cell receptor really emerges as the ideal target for antigen-specific therapy. So if we target molecules such as CD19, CD20, or even BCMA, that would lead to pan B cell or plasma cell depletion, and that would be a global immunosuppressive effect. But the B cell receptor is a surface immunoglobulin that dictates a B cell's antigen specificity. So that really then becomes the ideal way to do antigen-specific targeted B cell depletion. So the earliest report of CAR-T cell therapy, was published achieving complete remissions of B-cell leukemia and lymphoma, came out of Penn in 2011. And, we had always said in the laboratory, anything that works for B-cell leukemia and lymphoma is probably going to work for pemphigus. So when they started curing B-cell leukemias and lymphomas with anti-CD19 CAR-T cells, that really got us thinking about how we could re-engineer that design for antigen-specific B-cell depletion. So if we think about it, pathogenic B cells in pemphigus are uniquely defined by that surface anti-desmoglein 3 B cell receptor. And by definition, that's not expressed on a healthy B cell. So we replaced the anti-CD19 domain of a chimeric antigen receptor with a desmoglein 3 autoantigen, and we called it a chimeric autoantibody receptor, or a AChR. And that then directs antigen-specific rather than total B cell depletion. We showed proof of concept for this in various animal and in vitro models. And the hope was that if targeted B cell depletion with CAR-T cells is as effective as anti-CD19 CAR-T cells for B-cell leukemia and lymphoma, that could potentially lead to durable remission of pemphigus vulgaris after a single infusion without global immunosuppression. And this is now in clinical trials. I don't have I'm not going to go through the early clinical data today, but an ongoing phase 1 trial in mucosal pemphigus vulgaris has so far shown dose-dependent persistence of the CAR-T cells and no related serious adverse events or dose-limiting toxicities. So that then brings us to muscle-specific tyrosine kinase myasthenia gravis. This is an autoimmune disease, as you're aware, that causes potentially life-threatening muscle weakness. MuSK accounts for about 6 to 7.5% of myasthenia gravis cases. And like pemphigus, corticosteroids and B-cell depletion with rituximab are frontline therapies for MuSK myasthenia gravis. So David Richman's daughter was actually working with my collaborator, Mike Malone at Penn, who was an inventor of Tisagen Lec-Lucil. And, he was pointing out many of the similarities between MuSK myasthenia gravis and pemphigus vulgaris, which I found really intriguing. So beyond the fact that we treat them with many of the same therapies, including B-cell depletion and rituximab, the disease is caused by IgG4 antibodies. In the case of pemphigus, desmogleins are responsible for holding cells together, and the IgG4 autoantibodies interfere with that effect and trigger downstream effects. And in the case of myasthenia gravis, it inhibits the interaction of MuSK with LRP4. And just like rituximab's use for MuSK myasthenia gravis, the autoantibody titers drop after rituximab therapy. This is data from the pivotal clinical trial. In blue are patients treated with rituximab plus short-term prednisone. And in red is patients treated with high-dose prednisone. If you follow the blue line, it begins to drop, approaches the negative range of the assay. In the absence of repeated treatments of rituximab, it begins to relapse. But then once you start giving rituximab every 6 months, it falls into the negative range of the assay. And this data is important because it shows that the most, if not all, of the anti-desmoglein 3 antibody in pemphigus patients is produced by short-lived plasma cells that are dependent on that CD20 positive memory B cell pool for replenishment. And then furthermore, desmoglein 3 and MuSK share many autoantigen structural similarities. So desmoglein 3 consists of 5 extracellular cadherin-like domains. These are immunoglobulin-like domains. And MuSK consists of 3 immunoglobulin-like domains followed by a frizzled like domain. So we suspected that we could easily design the MuSK CAR structure to potentially fit within a AA CAR design. So we took the entire ectodomain of the MuSK protein, linked it to a CD8 alpha transmembrane domain and CD137 costimulatory and CD3 zeta domains. And we showed that this was robustly expressed on the surface of primary human T cells using an anti-MuSK antibody on the lower panel to show MuSK CAR expression on the T cell surface. And so we wanted to then check if this could then direct cytolysis, specific cytolysis of anti-MuSK B cells. So this is where we began to rely on our collaborator, Kevin O'Connor, who had cloned multiple anti-MuSK antibodies from patients. And he was gracious enough to share those sequences with us. So we have a NALM6 control cell line, which is which is BCR negative. And we can show that if you co-incubate these NALM6 control cells with either MuSK-CAART or non-transduced cell, we don't see any lysis, and basically on the y-axis, if there's killing of the cells, you'll see that line going up. But when we incubate it with anti-MuSK B cells, and for this we used a variety of antibodies, an anti-Ig1 antibody that was originally cloned by Martia Heibers and Jan Verschuren, an anti-Ig2 antibody, and an anti-frizzled antibody cloned by Kevin O'Connor, as well as an anti-Ig3 antibody that we made in a mouse. And when we express that on a NALM6 surface cell line, what you can see is that MuSK-CAAR-T cells robustly kill those anti-MuSK B cells, whereas non-transduced T cells show no cytotoxicity. One of the important things we wanted to look into was what serum autoantibodies would do, because essentially we would be infusing these CAR-T cells into a host with pre-existing autoimmunity to the therapy. And so the flip side is we also know that when CAR-T cells see their target, that can cause cross-linking of the receptor, which can cause activation and proliferation of that T cell. So there could be beneficial effects. And so the first thing that we did was we just did an inhibition assay in increasing levels of soluble antibody. And this was one where we actually took MuSK patient IgG, and we basically showed that in the absence of MuSK IgG, what you see is the curve on the top. Sorry, I don't have a pointer to be able to show that to you. And in the presence of MuSK IgG, that generally is inhibited, although you can get increasing cytolysis if you have a higher effector to target ratio, meaning if you just put in more CAR-T cells. We wanted to explore that a little bit more carefully, so we then incubated the CAR-T cells with increasing amounts of monoclonal antibodies. You know, we have to take this data with a grain of salt because we are using monoclonal IgG4 antibodies that are bivalent and not functionally monovalent. But in these particular circumstances, we did not see much inhibition with an anti-Ig1, Ig2, or Ig3 antibody. You can see the red curve is not really going down as you go to the right. And interestingly, we saw potentiation of killing with the anti-frizzled antibody. And we had also seen similar effects in our pemphigus studies, that some antibodies seem to inhibit, some seem to potentiate, and some seem to have no effect. We also incubated the, this is a mixture of monoclonal antibodies with the CAR-T cells, and we showed that these soluble antibodies cause dose-dependent interferon gamma production, which is a sign of T cell activation. And accordingly, we also saw that those antibodies induced MuSK-CAART proliferation. So the thought here is that CAART proliferation may ultimately counter any potential inhibition by anti-MuSK IgG. We then went into an in vivo model. And in this first model, we used a NALM6 model where we took our anti-MuSK B cells and put it into an NSG or immunodeficient mouse, and then tried to see if our MuSK-CAAR-T cells could kill that relative to CAART19, which is tisagenlecleucel, which is currently approved for clinical use, relative to donor-matched non-transduced T cells. And what you can see is that we engineered these cells to be bioluminescent, so we could follow them sensitively over time. And over time, these cells grow out. It will ultimately kill the animal. And despite equivalent disease burden at the time of treatment, what you can see is both CAART19 as well as MuSK-CAART cell treatment of these mice results in rapid reduction of the anti-MuSK B cells. Although I don't have time to show it to you today, that you will see that there are some mice that actually have a little bit of escape. This is not uncommon in some of these NSG mouse models. In the case of CAART19, it's due to loss of the CD19 antigen and or downregulation of the CAR on the T cell surface. In that particular mouse on the bottom right, it was due to loss of the BCR on the NALM6 surface due to outgrowth of BCR negative cells. We can never transduce these cells to be fully BCR positive. That is something that is an artifact, if you will, of this particular system. We don't think that that's going to be an issue in the human patients, just because the B cell receptor is the way that the B cell receives signals to activate and mature into an antibody secreting cell. So if the BCR downregulates, that's actually great because we think that that will render the cell functionally anergic. And if the B cell receptor mutates and no longer binds MuSK, that's also perfect because it'll basically render itself irrelevant to disease. So there's multiple strengths of this NALM-6 xenograft model. It is the model that was used to validate CAAR-T before it went into clinical trials. It allows us to test the human clinical product, which is human MuSK CAAR expressed in a human T cell. And that's sort of important from a drug development perspective. The anti-MuSK target cells we can engineer to be similar to those found in MuSK MG patients in regard to their epitope specificity, their affinity, et cetera. So very well defined. We can follow them sensitively over time. It allows us to evaluate CAR-T engraftment in this particular model because the NSG mouse is amenable to human T cell engraftment. And we know also that there is a high level of sequence homology between human and MuSK mouse, so that makes the model relevant to toxicology studies. However, there are definitely strengths. So this is a tumor model. So in some ways that's good because it's a pressure test for how well our CAR kills. The rate of CAR-T cytolysis has to exceed the rapid growth rate of the NALM-6 cells. But 100% of the target cells in this model are MuSK reactive, whereas MuSK reactive B cells in myasthenia gravis are rare. And ultimately, xenogenetic graft versus host disease will limit long-term studies. So we tested MuSK CAR-T efficacy in a syngeneic EAMG model, which mimics features of autoimmune pathophysiology. We modified some of the published data that was in the literature to immunize MuSK animals with a human MuSK ectodomain. The mice will develop anti-MuSK antibodies. The onset of symptoms is very sporadic, and I would say on the whole is rare in these animals. And then we treated them with either murine anti-CD19 CAR-T cells or MuSK CAR-T. For treatment. And then we followed MuSK autoantibody levels, total IgG levels, clinical symptoms, which really didn't appear in most animals, and then CAR-T engraftment. I will also note that when the clinical symptoms appeared, the mouse was generally dead within 12 to 24 hours. So it made it extremely difficult to do this study in symptomatic animals. So the advantage of this is that it has a lower frequency and number of anti-MuSK target cells, roughly 1 to 3% in this model compared to the NALM-6. And it has polyclonal serum anti-MuSK antibodies, just like human MG patients, and it avoids issues of xenogenetic graft versus host disease. And so what we did was, again, we immunized, we treated, and we followed them by flow cytometry. So here you can see an example of a flow. On the upper left are T cells, on the bottom right are B cells. In the CD19 CAR-T cell treated mice, we have no B cells after treatment, whereas in the MuSK CAR-T treated mice, there are still B cells that are present. And this was actually just more systematic data collected from the spleen and the lymph node. You can see that the black and the red dots are at the top, meaning that there's essentially a normal B cell number in those mice, whereas the blue dots are at the bottom, indicating almost total B cell depletion by the anti-CD19 CAR-T cell therapy. And when we looked at total mouse IgG in the serum, what you can see is that in the black and the red lines, that the total IgG level is increasing and normal over time, whereas in the CAART-19 treated mice, the total IgG level decreases and goes towards the negative range. Whereas the anti-MuSK IgG levels in these mice were decreased by both MuSK CAR-T and CAART-19 treatment, whereas the non-transduced T cell treatment had no effect. So this is antigen specific, not total IgG depletion in these animals. We did a number of off-target screens for MuSK CAR-T cytotoxicity, because this would be the first time that we sort of release MuSK from its normal expression pattern in the neuromuscular junction and basically allow it to circulate all throughout the body. We have no idea what it actually may interact with. So what we did first was just a series of unbiased as well as in vitro targeted screens where we just harvested all of the organs in the mouse, looked at serum chemistries and hematologic parameters in these mice, which we think is relevant because there is a high homology between human and human and mouse MuSK. And we did not see any evidence of specific off-target cytotoxicity in those histology analyses. We also screened a membrane proteome array that expresses over 5,300 membrane proteins. This is a great way to force expression of most of the known human membrane proteins, and it avoids problems with, for example, cultured cells losing expression of various human proteins. And we did not identify any hits on this particular screen that led to off-target cytotoxic interactions. We also did screen human primary cells or IPS cells and looked for evidence of pro-inflammatory cytokine production. We didn't see that. And finally, we did some targeted effects looking at both human and mouse myotubes that we differentiated in culture, and we did not see any evidence of off-target cytotoxicity or whether or not the MuSK could somehow integrate into the neuromuscular junction and just disrupt AChR clustering. So we didn't see any evidence of that in the targeted studies. So this then has led to the design of a phase 1 trial of MuSK in MuSK autoantibody positive MG. Just to hit the highlights, we're looking for patients who are adults who have active disease and they're anti-MuSK antibody positive. They should have been inadequately managed by standard immunosuppressive therapies. For major exclusion criteria, they should not have had recent rituximab. Prednisone is at a relatively low dose, and they should not have other autoimmune disorders requiring immunosuppressive therapies, because our plan is to stop other immunosuppressives cold turkey and to taper prednisone to a low dose prior to infusion. So just to give you an idea of how this is done, whole blood cells are collected from patients. The T cells are lentivirally transduced ex vivo with the MuSK. They're then expanded and infused back into the patient. Just before infusion, if patients are on CellCept or azathioprine, that will be stopped cold turkey, and the prednisone will be tapered to a low dose just before the infusion. And the initial part A phase of this trial will be a dose escalation. And the objective would be to determine the maximum tolerated dose and safety. I can't quite read the slide anymore of that particular infused dose. And part B will be an expansion to basically confirm safety and of the infused CAR-T dose. And so the primary endpoint would be related adverse events, including dose-limiting toxicities. We'll be also looking for manufacturing success rate, the persistence of MuSK CAR-T cells in the patient, as well as change in anti-MuSK antibody titer, and exploratory endpoints will be clinical disease activity. And so in close, this has been a way to bring forward precision cellular immunotherapy for MG. CAR-T cells aim to durably eliminate antigen-specific B cells while sparing normal B cells, hopefully leading to safe and lasting remission of B-cell-mediated autoimmune disease with a one-time treatment. CAR-T cell therapies are now being advanced for mucosal pemphigus vulgaris, which is currently in the dose escalation phase of the phase 1 trial, MuSK myasthenia gravis, and PLA2R membranous nephropathy, which is in preclinical development. And an ongoing phase 1 trial of desmoglein-3 CAR-T with lymphodepletion so far has demonstrated safety, no dose-limiting toxicities or related SAEs through 3 months in the first 4 cohorts, and has also shown a dose-dependent increase in desmoglein-3 CAR-T persistence, which is just now approaching the lower end of the range, which is observed for anti-CD19 CAR-T cell patients who had received prior lymphodepletion for treatment of B-cell leukemia. MuSK CAR-T preclinical studies indicate antigen-specific B-cell depletion without off-target cytotoxicity. And these data were submitted to the FDA and recently received clearance of the investigational new drug application. And fast track designation was also granted by the FDA, recognizing its potential to improve a serious condition within a serious disease with a goal of improving activities of daily living and muscle strength in patients with MuSK MG. And we're preparing now to enter clinical trials in 2022. I would encourage you to stop by the Cabaletta bio table if you're interested in learning more, as well as a poster number. I don't have the poster number off the top of my head, but there's also a poster covering a lot of the preclinical data that's shown here. It will be presented by Jinmin Lee, who actually was in my laboratory as a postdoctoral fellow and helped do all of the preclinical work that led to the approval. Not all, some of the preclinical work that led to the approval of desmoglein-3 CAR-T for mucosal pemphigus vulgaris. And then she transferred over to Cabaletta Bio, where she's now an associate principal scientist who helped bring desmoglein-3 CAR-T to clinical trials and is also helping to develop desmoglein CAR-T. I definitely wanted to recognize the individuals who are listed in red on the upper left, who are individuals in the laboratory that did a lot of the work that I showed you here today, as well as our MG collaborators and scientists who are in this audience. I've really learned so much from you over the past few years, and it's been a real pleasure to get to know MuSK MG and compare it to desmoglein-3 CAR-T. Thank you so much.
So it's noon, but we will delay lunch by about 10 minutes as we started later, and we have a long lunch break. So I think we'll take questions until about 10 to 12 past. Dr. Lawson.
Yes, please.
Please.
Was it to me a question?
Yeah. No, I'm sorry. I have a question for Dr. Granite.
Oh, please.
So in terms of your short-lived RNA-induced CARs, I think it's impressive that you haven't seen any significant CAR-T cell toxicities that occur with the longer-lived CARs. But I'm wondering a little, since it's not antigen specific, your CARs are directed against all the B cells in the body.
They are BCMA specific.
Right. But what I mean is antigen specific versus non-antigen specific. I wonder about the comparisons between the drop in antigen specific antibody with any associated drop in total immunoglobulin levels. As you increase the doses, are you going to produce suppression of non-antigen specific B cells and see the toxicities we see with generalized immunosuppression?
So the overall immunoglobulin levels as well as autoantibody levels are being explored. We don't have data to show yet. In theory, if you're able to wipe out all plasma cells, yes, you would get drop in, all antibodies, good and bad. So I guess the art in this will be to transiently inhibit as many cells as possible and then hope that the good, so normal, non-pathogenic plasma cells will regenerate. And depending on how and what happens in the autoimmune process, there will be a reset. I mean, in ideal life, right? We don't have the data, but if you could reset the autoimmune plasma cells, that would be the ideal case. It certainly has to be looked at.
I guess the potential
this is-
therapy might be then to combine both therapies to deplete the autoreactive B cell and get rid of the long-lived plasma cells and allow the long-lived plasma cell to regenerate from a healthy pool of B cells.
I agree.
This is Rich Lewis from Cedars-Sinai. To Dr. Nowak and Dr. Granite, the age-related differences in both the B cell depletion and in your small study of the 3 patients, it was interesting, your 3 patients, the 2 that seemed to respond were older and the one who didn't was younger. And we're finding with rituximab, the older patient population does significantly better than the younger population, which may have to do with the differences in the way our immune system reacts as we get older. And I wonder if we should be looking at these age-related differences more as we do these trials.
Yeah, so I think that, as has been noted, there's a lot of heterogeneity to response to a variety of immunosuppressive therapies. And even with the phase 2 rituximab MG study that we did, we found that there were certain groups of patients that actually had significant drops in their antibodies and others that did not. And so I do think that we sort of need to get back to the basics in terms of understanding the mechanisms of disease and understanding which patients might be more or less responsive to set treatment. And I think that's critical. Otherwise, we're just going to take a hammer and keep hitting the nail with these things. So be able to predict ahead of time which patients will do better on those therapies. And I think that applies to any treatment that we have. We can't get to the same place that MS is now, where there's a variety of different therapies, and which do you pull off the shelf for each individual patient? That really needs to be personalized to that patient, and we really need to understand the immune system and be able to test it in a way that allows for us to make those determinations ahead of time.
Yeah, I would just make the case that late onset versus early onset, we don't really understand it well. And there could be a huge difference in the way we respond to our medications, as well as the disease process in these late onset patients.
Agreed. I think there are distinct differences that we still don't have very well characterized.
So I will not reiterate all Dr. Nowak said, but I know that the sponsor for our study is looking at every possible biomarker in these patients. I mean, the number is so small that it could be just chance, but if there's a pattern that emerges, obviously that will have to be identified why certain people respond and certain others don't.
I have two questions to Dr. Granite. The first is you see some promising benefits in your, especially that weekly treatment group. You have 2 patients in that group. Did either patients receive eculizumab during that treatment? Because you said you allow patients to receive 8 weeks eculizumab, right?
I will actually call a friend for this. Dr. Quinn, did any of those patients receive eculizumab prior to the? So actually, I remember the story of one, although not my patient, and this from Dr. Howard. The person was a responder to eculizumab and stopped responding for several years now and stopped responding, hence the enrollment to the study. I am not familiar with the deep story of the other person.
Okay, the second question is, your treatment with the mRNA transfected T cells has a benefit that with the much the reduced side effects that often see in the DNA transfected T cell treatment. But the advantage of the DNA transfected T cells is long-last therapeutic effect. Sometimes it looks like remission. So in your case, did you track the stability of the CAR expression in your in vivo? I mean, in patients.
Yes. So the CAR activity. So in vitro, the CAR activity is about 7 days. If you actually put them with multiple myeloma cells, it's about 4 days. So when you look at these cells in vivo in a patient's periphery, within hours, the cells disappear. So I know the data from multiple myeloma. Again, the cells disappear, but then weeks later, actually, the clinical change starts. So there's an assumption they go into non-peripheral tissues, be it lymph nodes, be it bone marrow, et cetera. But that's by design, this is short-acting because of the toxicities that are a concern. So depending on the response, if there is a response, but if there is a response and good response, it's expected to be potentially transient unless we can reset the immune system. But if there is a relapse, the goal is to redose these patients.
Another question for Dr. Granite. I was wondering, since you're stimulating these or transducing these T cells with RNA, how they behave compared to the DNA transduction, because you might be triggering TLRs like TLR3 or 7 that you're not usually triggering with DNA.
Can you repeat? I could not understand what you asked.
I'm sorry. So since you're using RNA to transduce these T cells instead of DNA, I was wondering how immunologically speaking, they might respond a bit differently to that, since you could trigger intercellular TLR receptors like TLR3 or 7, which respond to RNA.
So I guess I should have put one of my disclaimers that I'm not an oncologist. So I'm going to ask the oncologist in the room to help me out on this.
Just take the mic. Yeah.
Thank you very much. Yes, we did look into that. And actually the cells do not upregulate their toll-like receptors, or intracellular nucleic acid rejection pathways in general are not upregulated after transfection with the RNA with the method that we're using.
Thanks.
I think we should let everybody go to lunch, and happy to take questions in person for anybody. Thank you.
taste it. Oh, taste it. But it's not forever. Oh, it's just for tonight. And only the greatest now. Yeah. Your sex is on fire. And so, my little darling, with the words to transpire. And so, your sex is on fire.